want, just John has a computer, so I guess there's, well, I guess someone in Zoom, so if you want to follow anything we're going to be doing, if you haven't done so, you have to subscribe to, you have to open Fiji, and you can subscribe to the neuroanatomy update site. That's just a temporary thing. SNT is supposed to be part of the new Fiji release, so you don't need to do that. Anyway, so in case you want to try to follow this, so the idea is that you type in neuroanatomy, click the checkbox, apply and close, restart, and then you should have this icon on your toolbar. I'm going to, the version I'm going to use is uses Java 11, so Fiji is migrating towards Java 11. It's kind of a limbo period right now. So my version may look slightly different than yours, but just details. Okay, so, okay, with that out of the way, and then, and then if you want to do accurate tracing, you probably need a mouse, like trackpads and gestures don't work well with Java's. Well, at least on Fiji. And then I was thinking, okay, so to start with, I thought it was perhaps useful to have like an historical overview of how SNT came about. And it's kind of, it's all like by coincidence. And so Simple Narrow Tracer was released in 2011. Janelia was involved in that, so there had been 2 hackathons on site, and Simple Narrow Tracer was, evolved also apart from that. That there was a PhD project from Marc Longueur. Then he left academia, and then the project was stalled. And then back in my postdoc days, I had the need to trace time-lapse, basically reconstruct dendrites in time-lapse videos. And there was really nothing out there that I could use. And so Simple Narrow Tracer was like this awesome, because it was easy to start hacking and start doing things. And so basically I kind of unofficially became sort of maintainer for Simple Narrow Tracer. And at the same time, I was doing also like these scattered plugins for neuroanatomy. The one based on Shoal was very popular, was released around this time. And then I joined Mouselight. So then I come here to Janelia. And then, then I had very specific needs. And again, the same cycle repeats. There was really nothing out there that I could use. And so I kept working on, in a very formal way, in a very kind of selfish way in Simple Narrow Tracer. So the things that we needed tools to prototype segmentation. We were trying to generate ground truth for machine learning. And so I worked a bit on that. And then I also needed, I was assembling at the time a library for morphometry. So basically, measuring cells, measuring projectomes and connectomes too. And then that was the point that, we officialized the effort. We created the organization morphonets.org. And then Kyle Arrington, now it's TZI, and Cameron, who was part of Mouselight, but now is at the Allen. So together then we kind of said, okay, we have this tool, we are doing all these things, let's put this together, make it, a formal package. And then, another key point also, there was, during the I2K meeting, there was the ImageLib 2 workshop by the Stephans. I participated on that, and that was kind of opened a bunch of doors. Cameron got all excited. We ported all sorts of stuff into ImageLib 2, and suddenly things just got faster, better, more efficient. I don't know if it's really because of ImageLib 2, but it had been like a legacy of 10 years that things had not been worked. But this was important because suddenly we could do things that we couldn't do before. And then we published SNT, I don't know, 2 years ago, I guess, 2021, I think. And then it has been really, really popular. And I think it's because the needs that we had are also needs that folks outside Janelia have. And so just to give an example, I was telling Dawn, so there is the neuromorpho database, which basically accumulates, all reconstructions that are published out there. They just ask folks for the STBC files, but they also ask the software that uses to reconstruct them. And SNT is like the most, I don't know if it's the most popular or the most, common. And I was telling Dawn also, it's interesting, like all the other ones, the top 10 are all Janelia software. So that's kind of, anyway, so now SNT is just this gigantic, weird, mega package for all things neuroanatomy. That comes, has lots of advantages, has also lots of caveats too, as you can imagine. That's a summary. So what I wanted, so basically, when should you use SNT, or why would you want to use SNT and not something else? And I tried to make a list here. The first one is that, it's part of the image ecosystem. That is, if that is awesome for you, great. If that is that you don't like that, then you probably are going to enjoy much. And then the other thing is that one thing that SNT does that is not very common in this kind of software for neural segmentation is that it supports multi-channel and time-lapse images, so up to 5 dimensions. That's kind of unique. And this, I think, has been really popular nowadays, with organoid experiments where people have neurons growing with other channels and, large volumes, light sheet imaging and so on. The concept there is that for the most part, everything is occurs in memory. Everything is loaded. For the most part, we only process, monolithic volumes. We don't chunk the data. Although some of the filtering operations, because they've been ported to ImageLib2, you can actually use them out of course. So that's kind of nice. But the tracing itself, the GUI, it's limited to stuff that fits in memory. The other thing that SNT does that seems to be quite popular is that, once you have traced your neuron either manually or some other approaches, you can use SNT to basically refine the tracings by snapping, the node coordinates to the center lines of the signal, and I'll show you that. The other thing is that SNT, just the way the GUI is organized, it's really not. It's very unorthodox the way you have to edit things manually, so that may not be your cup of tea. I mean, I'm grasping at straws here to find negative stuff, but that could be. And then the other thing also that SNT does, because it's involved, it's part of Fiji. If you have to ping pong between image processing and tracing, then it's awesome. You can stop, pause SNT, get SNT profiles out of the paths. You can convert them into ROIs. You can do all sorts of stuff. The consequence of that is that the UI is kind of generic. It's not very neurocentric. People use it for segmenting, microtubules, all sorts of filamentous structures. And so that, if you are an aficionado, if you are really interested in, neurons, you may find that off-putting. And the other thing is that, there are no black boxes. We try to be as transparent as possible, but that the consequence also, there's no big green button. So you have kind of to explore things around. And then the other thing and this was from day one, kind of again, by coincidence, but also by need, it's all about scripting. So you should be able to script every single thing, including the tracing also within, of course, some constraints. The cons there is that if you want to do it in Fiji, you actually don't have really, it's not IntelliJ, it's not an IDE. So you probably will do it from the script editor. We try to improve the script editor a bit, but some things you are going to feel that they are not as powerful as you wished. And then the other part is that if you want to use it as a library for Morphometry, basically measuring all things about neurons, and it's awesome. We are convinced that we have the most comprehensive library with built-in functions for measuring all things about neurons. This is better done with proper Python, with native Python, with PyImageJ. That can be finicky at times, but that's a trade-off. And then again, as I said, it's very popular, or it seems to be very popular right now. So there is a large user community around it, but the developers is just pretty much me and Cameron, and in part-time and fading out. So anyway, so I wanted to show from folks here at Janelia, I have been receiving either on Slack or email messages about SNT. So I selected what I think what are the most common things about. But if you guys want to show if you guys want to discuss something else, we can, just interrupt me and we can do it. So the other functions, well, the easiest way to access SNT is through clicking the button. You get this shortcut dialog here. You start that console here.
Tiago, if I can interrupt you.
Yes.
You're clicking buttons really quickly, and I don't always see exactly which ones you click on.
Oh, I see. Okay, I'll do it much later.
Say what you're clicking on.
Yeah. So I click this button. These are basically all the entry points to software. The main one is SNT proper. The other ones are viewers, in case you don't actually want to trace, you just want to visualize data. And then these are access to other plugins that you can use without having the full SNT open. So the dialog, is typical ImageJ. It's, not fancy at all, very simple. And And it was very funny. So one of the reviewers one of the concerns he had was, this is nice, I can understand this is powerful, but it's Fiji, and Fiji is super clunky, it's ugly, I don't understand, it's like, it's super crappy, it's hard to use. And so literally we spend most of the reviewers trying to prettify things. And so I'll show you, the reason I want to show you these is because if you are doing something for Fiji, please use this, because it's nice that these things, because it is true, Fiji is very, it's very 80s. So, so we can bring it to the 90s maybe. So anyway, so what we are using, so it's written in Swing, the GUI for, but there is a library out there. So Swing is still a thing right now. There's a library out there, it's called FlatLab that, keeps patching all the bugs that Swing has that has not been updated in many years, and they make it, scalable icons, SVG icons, and all sorts of stuff. That's what we are using here. This part now is part of Fiji, and so we try to make, so out of the box you get customizations, you can move things around. That has not been always a tradition in Fiji, but we try to make things user friendly. So these, the sections are divided by headers, and if you click these, you go to the manual. I know these are, minor details, but
Awesome.
yeah, but it just makes things accessible. And the other thing we did also, we have, you have utilities here, a command palette, and this is typical what you find on, you can type for things and you can look for them, and it tells you where, so if you want to, load something, you can look for them, and then you can click, and then it opens. And so that's one thing. The other thing we did also, there is, we can access actually from here. There is a script recorder. So basically, not all, but most commands that you do are recorded. And the function here is not really to have, fully fledged functional code. There's two things. It logs what people are doing so that they can remember, and reproduce across members in the lab typically. Or if they're doing a bug report, they can say, look, this is what I did. And that seems to be useful. And of course, some people use these as a starting point to then, script their stuff away. So we try to make it, again, because we were criticized because we were not fancy enough. So we have some demos here. These seem to be useful. So basically, we try to have different data sets for different types of functionalities. So these are going to be, multi-channel or a time lapse. Of course, these images are very small. They are downloaded from the internet. But, and then you can look for them here, different demos, and you have a description here, what the data is, if there's a paper associated with it. So you can start with this one. That's just a silly toy neuron. It's nothing really special. But I'll show you. So here you have the main functions. Here you have the options on this tab. And here you have the viewers. And the viewers, this is historical. We have our own viewer that is a 3D canvas. We can do stuff. It's just only allows for neurons and meshes. It doesn't allow for images, so it's just a visualization tool. We have side view, which I'm not going to show you today, but that's where Kyle join in. So side view is the becoming powerful 3D viewer for ImageJ. It supports VR, it supports meshes, images, masks, so on. But it's still very, it's still, it's not in a mature state yet. And then we have the legacy 3D viewer, which has not been updated in 15 years, almost, 10 years. And so we keep it here for legacy reasons. And then you have the other key component of SNT is the path manager. This is equivalent to the ROI manager you have in Fiji, and this is where you can do all sorts of stuff with paths. It stores them. You can assign text to them, so you can label them by which channel they were traced on. You can, but importantly, you can actually do things with them. So you can filter. So for instance, let's say if you want to look for all the paths that are between 10 and 100 microns, so it selects, and then you can delete them, tag them, and this is useful for folks to, curate annotations and so on. Anyway, so in a nutshell, that's pretty much what There are other functions here. There is, well, you can create dendrograms also. So you can, if you're doing graph analysis, you can do. There's a viewer also to render, flowcharts and diagrams and stuff like that. So let's start with the multi-channel image. So I'm going to load the demo number 5. So this is, I actually don't know where this image comes from. It's either comes from the cell image library, I think. So it's a multi-channel image, has 5 channels. We can just select one, so we can trace on the right channel. So you would select the channel here if you want to trace channel 5. In this case, we're going to trace channel 1, so it's already set. And then, it's a semi-automated tracing. The way you do this, you just click on the starting point, click on the end point, and then there is an A star that, finds the 2. There is no undo in the program. That is, that's kind of a bug, but we make it a feature in the sense that we think that by having it so, if we force people to be disciplined when they are tracing. Anyway, and then there is this prompt here that is going to ask you all the stuff. So you now knows that you did a segment. So he asks you, what do you want to do with the segment? And you see that the shortcuts, you have these letters that are highlighted. Those are single cell shortcuts. So if I want to keep yes, I just press yes. If I want to finish, I press F, and then it's done. And then if it's these too annoying to you, you can just, remove all these options, and then when you are done, it just finishes. Yeah, so you see also here, there is this shortcut here I highlighted. So if I want to toggle filter number 1, I just press 1 and so on. So that makes it easier to know the shortcuts. Okay, so that's pretty much it. Things that may interest you. So there is, to improve the accuracy, there is this cursor snapping. So basically, you define a neighborhood in X, Y, and Z dimension, and then the cursor looks for the brightest voxel in that neighborhood, and it also clicks there automatically. The core of the search is the A star that Mark still had in the original Simple tracer. We just did some minor modifications. And then we wrote one it was published more recently, which is called NBA, stands for New Bidirectional A star. And that is, depending on the images, can be faster, but I have to say the results are very similar. And then here you have options to, adjust the cost function that you're using for tracing, and you have to read the documentation. For this, one of them was taken from Horta, because we were happy with it and worked with our images when we were doing the ground truth data for the machine learning. Others are taken from, literature, and others are kind of made up, actually. One was just kind of made sense once, and then we start using it. And then you have options to how do you want to do the computation? Do you want to do it in real time? So this means that every time you click the starting point and end point, for that bounding box is going to be computed, or you want to do for the whole image. And this used to be a thing, when we started, this was important. Nowadays, with modern computers, I don't think it really makes a performance difference. Okay, so that's basically it. So, so that would be, so then you have a path. If you want to change channel, you can go now to channel 2. You trace the channel, now you get different channels. Okay, so that is, I think I've showed you this. Okay, I'll try now to load another demo for a different data set. So I'll use, this is from the diadem data set. This is the most common image people use to benchmark algorithms for neural reconstruction. It's really popular. It's kind of sad that, it's almost, 15-year-old image, and this is still today what everybody works with. But, it tells you the importance of having really ground truth data sets. And this is literally the only one for fluorescent image so far that people use. Anyway, so SNT doesn't really support 3D, but it does these so the way you work on 3Ds through these orthogonal views, where you have the XZ and the ZY view, and then basically when you are browsing your neuron, you can, well, I just synchronized, you press shift to synchronize all the views, and then basically you can see where you are in the different axes. And I can, if that's confusing, I can toggle that. I can just put the volume is not very isotropic, so the side views are very narrow. But I'm going to.
So it does 3D, but it doesn't do volume rendering.
Exactly. Yes. Nice. It's true. Yeah. Well, thanks for that. So what I want to do, okay, so going back to our to-do list, so one thing that SNT is pretty cool to use for is if you have, let's say, you have a I just got an exception, 2 exceptions. This never happens. So one thing that people use it all the time is if you have some routine that segments neurites, but it doesn't, just reconstruct, just obtains the center lines, and you want to obtain volume, you want to obtain the radii along the neurite. So you can use what we call path fitting, and I'll show you, so you have to specify some parameters. And so basically you can choose what you want to do. So basically the idea here is that you have an initial center line. You can do 2 things. You can snap the signal back to, to the maximum along the signal, or you can assign radii from the cross section of the signal that was, associated with the original path. So you need to establish a maximum, radius that you are willing to explore to make sure that you don't have, crosstalk from neighboring neurites. And then you have to define, what you want to do if something fails. Do you want to default to some constant? You want to obtain the best guess, which is like a mode or an average of the flanking fits. And then you also have to define, the minimum angle between, the orthogonal position from the previous. And you'll see that in a second. And then you can decide also if you want to replace the existing nodes, or if you just, have a fitted version of your path, and that is just a mirror copy of the data, so that doesn't override. Okay, so I think right now, so you can just use the default, and now you can. Here's a demo to show you. So it's telling you that you should go. It's the first time that you're running this command. You should go and visit the manual, and then basically, so basically you have here the green is the original position. The green, the diameter is the mode of all the fits that were done at the location, and then the yellow is the adjustment, okay? And then you have debugging, you can see the angles from the previous path. But this has been quite popular. People are using this quite a lot for, when they just trace, center lines or skeletons, and then they try to obtain. So this has been, I'm actually quite surprised that people have been using this a lot. And you see, like, sometimes there's a failure. In this case, the displacement was just too large. And so, it gets marked as red, and then, the default choice that you had kicks in. So that's kind of, that's a neat thing that SNT does, and I think most, that offers some flexibility to do this. Another thing that, okay, so that is, I think I'm going to delete all the paths now, and I'm going to, why is that path still there? Okay, I'll just start afresh. So I'm going to do the same demo again. Okay, I'll just. Okay, so now we have back the same image, and now I'm going to pause SNT. When you do this, you basically wave back the image back to image A, so now you can just use it as you can do any operation you're going to do. And now, this image is really, it's a grayscale image, but you see the signal to noise ratio is really exaggerated. The background is pretty much pitch black. So I'm going to degrade this image a bit. So I'm going just to introduce some noise. that's just not enough. I'm going to make more noise even just to make the point. I don't know, I guess some salt and pepper would work. Okay, so now you have an image that is graded. Okay, so the image now is dirty, and the signal noise ratio is worse. And let's say you want to trace the main accent. Oh, now it's paused, so I'm going to unpause it. You notice that the image changed since the last time it was loaded, so you can, you reload the image, goes back, and now I'm going to trace this path here. Okay, and it's much slower now, and I'm just going to finish. Okay, so this kind of data. So the idea is that one thing that SNT does is that you can trace by layers, and so that's what I want to demonstrate you now. So you have this wizard here, and so the idea of the secondary layer is that you always have access to the actual image in its native, SNR. And then you can trace on a cached version of the image that has been preprocessed. Okay, so we have, for these we are using image AOPS. Some of them, the tubeness, we extended to be a multi-scale filter, and we did some modifications. So that's the default. I'll just use that in the default. And so you have, and then the key criteria here is that you have to define the sizes of the neuroids that you are tracing. You can do that in two ways. The easiest way is to do visually. So, the wizard now is asking you to click on the representative structure. So I'm just going to click on that location there. You have to wait a bit. Okay, and now you have a preview canvas of the different, so this is the preview of the filtering operation, at the different scales. So basically the sigma, and then, you literally just choose the ones that you think are important. So in this case, I would probably choose these. I'll probably just, move up and down to make sure everything's okay. You can also choose this one, that will be okay. And then in here we have controls to adjust the scale. So I'm going to commit these 5 scales. Okay, and now you can decide if you want, if you have enough memory, do you want to do the entire thing? Or do you just, compute as you go? In this case, I'm going to do the entire thing so that we can look at it before, but you actually don't need to look at the image. Okay, so I'm just going to run this thing. You have to wait a bit. Again, this will be faster if you were just doing computing as we go.
And all this processing is happening locally?
Yes. Come on, man, don't die on me. What happened? Okay, so that's how the image looks like. So it's way better, in the sense that, neuroids are now smooth, the background is, has been minimized. And now you see that you now have this option here that, you can trace on the secondary layer. The shortcut is L. Again, it's highlighted there, single key, so you can just press L. And now we can repeat the same tracing, and then we can compare the result. So I'm going to trace the same path. It's faster, of course, and you can compare one path or the other, and you see that one is much more smooth or natural, and the other one is more. And the cool thing about here is because this is going to work on some places, not going to work in all the places, but you can toggle this at will, right? So you can. You can try, if you don't like the result, you can toggle the layer, just press L, and then you trace on the raw data, and so on. You can overlay the filter version 2 to have some, enhancement. In this case, that's, you could make it, 40%, I guess, or something. So now, this is just in the image overlay. You are still tracing on the raw data. So that's kind of another neat feature. And also surprisingly, people have been using these just to generate filtered images that you can then threshold, or you can do other stuff with it. So that's kind of, so these, in retrospect, some of these things should move proper image AOPS. The reason why they didn't do is because at the time we were not certain if we were being accurate or not. And so we decided to have them in SNT and not migrate them. But probably now is the time to move them. Okay, so I think we've done that. Okay, other things that people use SNT a lot is to convert, tracings into an ROIs and then do image processing with them. One thing that you can do is, for instance, you can use trainable weaker segmentation. You can train a random forest classifier from tracing. So the idea here is that you can trace one or two paths. Let's say you have a complex neuron that is going to take a full afternoon to do it. You can trace two paths, use that to train random forest classifier, load that into a secondary image, secondary layer, and then trace more quickly. Kind of that's the, that's a possible workflow, or you can just use it to train, to get a mask or a labels image of sorts. So in this case, I have to say it's a bit clunky, but it works. So you need to have some, so if you are training a random forest classifier, that you need to have two classes, right? You need to have the neuroids and the background. So for the neuroids, you use the paths that you traced. For the background, you need to use ROIs. So the way to do it is that you pause SNT, and now you just do some scribbles around the background, some ROIs, and then you store those in the manager, in the ROI manager, something like that would work. And then you can go here, run this command, and then you can, so you can obtain a labels image or probability PMAP. And then you can decide what you want to do. You can look, and then we can just load this straight as a secondary layer. And I'll can do that now. Hopefully it won't crash. And this is actually a script. So you see, like, there is nothing sophisticated about these because, but it's just a convenience. And so most of these commands are actually scripts that just call, different APIs and just try to do everything. So you have this actual convenience to do stuff. We need to wait.
In the ROI manager, are those the things that you traced in the background that you just kind of?
Are those yellow? Yeah, the other
little things you flipped into the background.
Yeah, I think not.
Okay.
Because you basically need to have something that marks what is background. The absence of, you could make, it could make everything that is outside the trace path, but that is not going to work because you could have neuroids that have not been traced or something else. And then,
okay,
this is taking forever. You have to kind of trust me that this works. This is a loaner. I actually never use, I haven't used Mac in a while, so I don't, on my computer, this runs really fast. So anyway.
it has a little status bar up in Fiji itself.
Yeah.
Okay.
Okay, so it's telling you, so now this is your secondary layer, and you see, like, it's kind of, it's usable. So now the idea is that you can make more natural, curvatures, and it should be much faster now to trace things. And I think I don't have anything else. There are all sorts of stuff, so I just don't know. Again, these, I think, were the most common things that people ask me. If you guys want to see SNT to do morphometry stuff, we can do it, but I just don't know if that, you guys,
Yes, from me.
Yeah, okay, so, I have to show you something. Okay, so that's basically pretty much, that's the tracing thing. Again, so things here that you can do out of core are the filtering. You can do it, you have to use script for that. The GUI does some of it, like the secondary layer, you can compute you can use compute as you go, but the main image will still need to be loaded in memory. So it's kind of, again, you cannot do this so far for like really large volumes, but you could. The infrastructure is there. Now it's all these, random access intervals, all image data structures, so you should be able to use it. It's just, I don't have the bandwidth to do it, and I don't think Cameron has, but, someone wants to use it. And sorry, I'm trying to think, is there anything else? Then, yeah, okay, so we can do, okay, so I'm going to close everything now. And I'll just show you now SNT as a library for analysis of neurons, of reconstructions. And I guess we can close the PMAP too. We can close all these images. Unless you guys want to do, I know, is there anything else? Oh, I forgot about, so there, there's the field manager, which is equivalent to the path manager that for fields, and that's to generate masks. So basically there you can generate labels, image. So basically every path gets a unique label, or you can have a binary mask. That's super useful if you want to do image processing routines and you have a complicated, you have a filamentous structure that you want to use as a mask and you want to have a quick way to do it. SNT could be used for that. The workflow there is that you trace a path, you open the field manager, and then you generate a mask out of it. Why there is a field manager and not the path manager? Because we were not sure about the field manager would work, and so we created a different thing on the side, and then it just stayed on its own. Okay, so I have to show you. So okay, so let's say. So the idea is that let's say you have an SWC file. I guess this one you can just drag and drop there. Okay, that's probably not. So the idea is that these are your entry points to other different aspects of SNT. So you have the reconstruction plot is just a 2D viewer of a neuron. And so in this case, this neuron is 2D, so it automatically loads. There's nothing special about it, but you can zoom in, you can do stuff. You can export as an SVG. That's useful to make figures. And then you have, the more mature viewer, which is this reconstruction viewer. And I can show you some examples. So you can load, you can drag and drop stuff there. I was just going to load some. In this case, these are mouse-like neurons, so you can load. Neurons manually. You can load mesh manually, or you can connect to the major databases. So right now we can connect to Fly Circuit, insect brain, mouse-like, neuromorph. Neuromorph is like this umbrella, so they store pretty much all the others. So it's another entry point. And here the reason why I have this dedicated viewer is for two reasons. We wanted something that it was easy to use and something that would allow us to do quantitative visualizations. And I'll show you what I mean with that in a second. But just an overview. So these things also has single key shortcuts. You can toggle dark mode, you can toggle axes, you can make animations, you can zoom in, zoom out. All the stuff that you kind of normally would do. I know it's unfair, but you probably don't see what I'm doing, but I just know the shortcuts. And so, you can load meshes, you can load reference brains. And that's what I want to show you because I think it's where is it? So you have the different fly brains, and there is an Easter egg here. Yeah, you've seen it. Oh, why is it not appearing though? Oh, man. Oh, yeah.
Yeah. Is that your brain?
That is just my brain. There's a fly. There's a little fly inside here controlling everything.
Yeah, so I always love to do these. That's the fly brain right there. Oh, it's very bad on the screen. Oh, this looks like crap here, but do you see that dot there? That's the fly brain, that's the mouse brain. And we can load the zebrafish brain too. The zebrafish. And that's that bigger dot there. So anyway, okay, so maybe we can do some, so this has the recorder too. You can use the recorder, but I guess you guys probably will never use it. Again, there's a script recorder here too. You can say, you can record the script and you can do stuff and then it will log here and then you can use that on your screen or you can just start one of yours. Okay, I'm going to close all these. And let's do this properly. So let's just start with the script. In this case, you do template, let's do Python. Then you open okay, and then the templates are just, you have a bunch of imports, and so you don't need to worry about them. Right now, it's scripting in the script editor is all a bit clunky. Auto-completion only works with Python, which is Jython, which is Python 2.0. Debugging, it's nice in Groovy, but you don't have auto-completion. So, and then, I was telling about, the grumpy reviewer. So like we did some things here to make it better. So auto-completion, that was done with Albert Cardona. That was nice, but you also have the command palette here, so you can look for stuff, can look where commands are. And then more important, all our scripts, so we have like 59 or 60 something scripts, and they're all splitted here. And so like, these are all here under neuroanatomy. They are divided by categories. You can look them by language too. We try to make as many languages as possible. So, if you go for neuroanatomy, those are all the Python, you have Groovy also, and so on. And then we also try to make these, we don't succeed all the time, but we always try to make the demo scripts here that you don't need to type in any data. So if, so in this case, I don't even know what this script does, but we need to look at it. But, for instance, you can do, let's say, the render. Yeah, okay, so there's this demo, you can try to run it. And so if there is, so in this, so the quantitative visualization, what I meant is this kind of stuff where you can color code morphometry into your scene, either into the meshes or the reconstructions themselves. And then, you can, use these as a visualization. In this case, in this particular neuron, the dendrites are in yellow, and here it's, I think it's mapped to distance from soma, and then those are the primary motor neuron, primary motor area in the cortex area. That's maybe too specific for here. Okay, so the way you start, let's say you want to do something. So the starting point, let's say you have an SWC file, and I'm just going to follow up my notes here. So the starting point, a reconstruction in SNT is a tree. So you need to initialize a tree from, typically from a path, if a local file. So in this case, I'm just going to go to my demos and then just get, I don't know, some, guess this neuron. You should be able to drag that. Oh man, so much for user-friendly. Okay, so now you have a tree. And now auto-completion should kick in with control space. And now you can, if you want to see how it looks, you can try show or show 2D. Show 2D shows the neuron in 2D. If you do show 3D, it does show the neuron in 3D. Okay, so in this case, you see like the color is, you don't see anything because it's just a very default gray. So you can change the color of that. You can just use auto-completion. And you can look for color. There is one that uses string arguments. We can just type red, and this should work now. So now the news. Okay, so in a nutshell. So the idea is that we try to make it, because we tailor these for Python notebooks. We try to make that make it. I don't know what the term is, like Pythonic or simple, that to do stuff. So, okay, so the starting point is a tree. Unless you want to do really sophisticated, graph analysis. In this case, you would have a direct graph, or you would have, or if you're doing something like persistence homology, which is more complicated, then you have different objects. But for the most part, you would have a tree, or a group of trees, or a collection of trees. And then, if you want to measure a neuron, you would probably do. You need what we call a statistics instance. So in this case, you have a tree, you have tree statistics. If you are looking at nodes, you have node statistics. If you are looking at groups of trees, you have group tree statistics, and so on. In this case, we just have a tree, so we just initialize the tree statistics. And you do it from a tree. Okay, so now you have a statistics instance. Now you can, do stuff with it. So let's say, for instance, if you want to obtain, we try to make it simple so that folks can always obtain the reason why I needed these is when we are trying to explore, different brain barriers in the mouse-like data set, we wanted to have an interactive visualization, try something, then render it, and so on. And so that is reflected here. So for instance, there is a function here. Let's say histogram. So let's say you can obtain an histogram, let's say for internode distance, which is not, kind of a boring metric, but you can press show, and now you have an histogram, and they look nice by default. There is some fuzzy matching here, so things should work if you type, before failing, if you type, internode dx, kind of a standard abbreviation for distance should work too. Okay, so now that's basically your tree statistics. So now if you want, I'm just going to copy. So if you have a collection of trees now, you would do, you obtain the trees like this. In this case, I'm just pointing into a group. You initialize, now you don't initialize tree statistics, but you initialize multi-tree statistics because you have more. And then the cycle repeats, it's the same thing. So now you can obtain, you can obtain the same histogram if you want to. Oh man, I cannot type. And then we can do the same thing. We can do show again. And that's not very useful, but now you have an histogram for different. Oh, there's power histograms. Oh, I see. That's nice, actually. We use this for angles. The idea is that without minimum effort, you should get some reasonable outputs. Like you would, yeah. And this was a direct answer to Adam Taylor. That's always saying that he uses MATLAB because MATLAB just makes things nice out of the box. And like, duh. Anyway. So if you have a collection of, let's say if you want to compare populations of cells, then you have group tree statistics, and then the same mantra, the same thing. You have a collection. Okay, I'm just going to run the snippet rather than talking, and then because now the type of knowledge is different, so now your histogram just automatically separates the two populations, and you have group one. So in this case, we just loaded. So initialize the instance. You got a collection of trees. You assign it to one group. We named that group group one. We open another folder. We name that group group two. And then, we can do an histogram and they are separated, and you can do box plots and stuff like that. And there's more sophistication to it, but, I think that's much really too in the weeds for, type of analysis that I think you guys would be interested in, unless you are really passionate about knowing, how many branch points are there in the subiculum or on layer 3 of secondary motor cortex or something. So in a nutshell, that's it. So there is modeling also, and that's from Cortex 3D. So that was a project that had been abandoned for a while. But it was written in Java. So it's a simulation engine that allows you to do simulate morphologies. So that is incorporated. That was the work of Kyle. That's incorporated for SideView. So you need to have SideView installed for that to work. And I think in a nutshell, that's pretty much it. But again, this stuff is, people have been using these more from PyImageJ with Python notebooks. Not really, yeah, because again, the script editor is kind of, people find it finicky. So I don't know, there is, if you guys want to,
People prefer the Python notebooks over the script editor
yes.
because they help more.
I don't know, I think, I don't want to sound silly, but I think it's more, I think most of, the audience for SNT outside Janelia is the typical, smaller lab that studies cell biology of neurons. I think most of the work is from undergrads, and there's like, oh, you need to use Python notebooks. I don't know. I think all the questions I have is all about Python notebooks. So yeah, it is. I mean, it was a So we have a series. So if you go on the help menu, we should probably make this better, but we have a series of tutorials in Python notebooks. And then you have, if you are able to install everything to start with, so you have basically what we talked about. You have more basic stuff, and there by order. You have number 1. Number 8 is more sophisticated, but it's cool. I mean, number 8. I think. Sorry, I don't know which one it is. Yeah, okay.
Yeah, that's the one I want to see.
The persistence landscape.
Yes.
Oh yeah, it's cool.
Is the code for this again SNT probably, right?
Yes.
Yeah.
Yeah. So this is clustering using, these here. We basically take a population of cells that are in the same place, but they go to different places in the brain. Then we use precision homology to segregate them. We do clustering, and then we separate them. It's a bit unfair because we picked those because we know we could distinguish them, but it was unbiased. And then there is another. There is one. I don't know which one it is actually. I should know. That we use Blender to render actually. And that was nothing fancy about it. It was just because, then we can showcase. I mean, since we're using Python, not just. This one tries to distinguish, if neurons have a bias towards, ipsilateral or contralateral hemispheres. Again, that's probably too much in the weeds for you guys, I think. But these are graphs out of the box. So it's, you should be able to get decent. So I don't know where the Blender notebook is. Where is it? It's this one, I think. Or is it the convexo? I guess I changed something. Oops, that one is not. I think it's this one. So we do convexal analysis too. That's using ImageOps2. And in this case, we are measuring convexal looking. The question here is, what is because on the type of data from MOSLight, we don't have synaptic strength, right? We just know where neurons go and now how much they innervate. And so, the idea is to get surrogates for, the impact of a neuron versus another. So in this case, we are measuring the convexal of all the axonal terminals, and see, what's the influence of a neuron in a certain area of the neural peel versus another. And so I think the Blender file is not here, but anyway, we have a Blender somewhere. People like it, but they hate installing. Everybody freaks out with installation of PyImageJ, and then you have the imports are clunky because you have to use, the Java classes. You have the camel case thingy. People get upset by it, but once they have it running, then they don't complain. But the script editor, I think the reason of the script editor is, I don't know, but Python 2 is annoying because, people have, they have, some things are different, the parentheses and stuff, and so people just stop writing and then they don't, they say it doesn't work. And then the debugging is kind of, so the auto completion is nice, that was our pretty much all of it, but the debugging is bad. I did that part. And so basically when you have an error, when you have a line, that errors, because, the script editor populates all sorts of stuff before with add parameters and all that. So there's like a huge offset. And sometimes for Python, that thing never works very well. So, it tells us line 20, but it's actually line 14. So that can get on your nerves quite a bit, easily. Groovy was awesome. Groovy is nice because it errors, tells you which one to line, and even suggests, are you sure you didn't mean to do something else? But it doesn't have auto-completion.
No, I was just wondering because, but from the Python notebooks, you get like Python code completion and stuff like this, but you don't see anything useful in the Java classes or stuff like this when you want to use PyImageJ to call anything directly.
Yeah, that is true. Yeah, that is true. I think in this case, people just.
Right.
I think people just use the examples that we have, and so then So we brute force our way through documentation, basically. I mean, it was just every time someone asked a question, well, Cameron, when he had more time, he would just then write a script or a Python notebook or something like that. So that's, in a nutshell, that's, it is super popular. I was impressed how popular it is, actually.
Yeah.
Do you know what is, sorry, I was telling Don, so the neuromorpho guys, they collect reconstructions from, every time there's a paper that mentions an SWC file, they contact the authors and they get the file. And so they keep a tally of the software that was used to reconstruct. SNT is like the, I don't know, 90% of reconstructions are done with SNT in a way or another. But all the other ones are all Janelia software. It's super cool, And then there's like NeuroLucid at the end or something. So it's kind of cool, yeah. And then people cite the Fiji paper.
That's annoying.
It's like we reconstruct neurons using Fiji. Hurrah. So if you want my opinion, I think this is the, SNT is the only reason why Fiji is successful.
Probably right.